very pleased that Professor Penny Moore is giving today's IDORI seminar entitled Pushing the Envelope, HIV Vaccine Discovery in Africa. Penny has been providing valuable insights to me and IDORI since its earliest days, and she is the Sarchi Chair of Virus Host Dynamics and Research Professor at Wits and is at NICD. She's director of the Antibody Immunity Research Unit. It's an extramural unit of the South African Medical Research Council. And she holds a joint appointment as honorary senior scientist in virus host dynamics at CAPRISA at University of KZN. With funding from a variety of donors and very strong focus on mentorship and capacity development, Penny's very productive research centers on humoral immune responses, especially neutralizing antibodies, and their interplay with viruses like HIV and most recently SARS-CoV-2. So very excited to welcome Penny Moore.
So our unit has for a long time worked on HIV and then SARS-CoV-2 and then subsequently other viruses. Any virus that mutates and can get away from antibodies is interesting to us. And in parallel, we study the antibodies that push the viruses to escape. And it's kind of an endless arms race between the two. Both evolve as fast as the other. We are seriously antibody addicts, as you'll see from the picture here of the lab, where everybody was trying to make those Y-shaped molecules and generally trying to give me as hard a time as possible. But this is the crowd who's done most of the work. You can see it's a lot of students, a lot of people, a lot of Witsies with master's degrees who work as staff scientists. It's a really wonderful crowd of people, from honors students through to PhD students and onwards. The work that we've done in HIV for a very long time is very much studying HIV where it matters. Rob mentioned that I'm part of the CAPRISA consortium, and CAPRISA is the Center for the AIDS Program of Research in South Africa. And it's a program that Slim Karim, who's shown there, started 25 years ago now, studying people in almost all women in KwaZulu-Natal at two sites of Vulindlela and Durban, which are shown here. And the reason I say it's studying HIV where it matters is because in that part of South Africa, in many parts of South Africa, but specifically in that part of South Africa, the rates of HIV are still crazy high, still up at the levels above 50 or 60 or 70% if you go to antenatal clinics and look at young women, particularly that group, at huge risk still in KZN. Men less so in that community, but the levels are really still spectacularly high. So this is why we have spent a lot of time in that area. So it's still clearly, as everybody in this room will know, a major public health issue. Undeniably, ARTs has made a huge impact to the management of HIV. And that's particularly true very recently with the lenacapavir data that came out a couple of weeks ago, I guess now, which I think is going to change the HIV landscape completely. But I still think that a multifaceted approach is wise to pursue in the context of HIV, and that for me includes an HIV vaccine. We also do some work on HIV cure, though frankly I'm less optimistic about that. But the work I'm going to talk to you about today is largely thinking about HIV vaccines. So why is it so hard? It's a question that I get asked often. Why was it so easy for the virologist community to come up with a COVID vaccine? And many of the tools that we used to develop those COVID vaccines came out of research that had been done in the HIV field. But I think the challenges are completely different and unchanged actually for HIV. The HIV particles are almost completely naked compared to the SARS-CoV-2 particles, as you can see here. So the envelope that we're trying to push is those little blobs on the outside of the HIV particle, and you can see they're few and far between. In fact, this one's probably generous. There's somewhere between 6 and 8 spikes per particle, so it's difficult for the immune system to see it, whereas SARS-CoV-2 is laden with spikes, and so it's really easy for the immune system to see. The SARS-CoV-2 spike is somewhat glycosylated, and of course glycans reduce the visibility of the spike to the immune system, particularly talking about the antibody system. Whereas the HIV envelope glycoprotein is a bit like a Smartie in the sense that it's almost completely covered in sugars and nearly impenetrable as far as the antibody response is concerned. So it's very, very hard for the immune system to see it again. And then the variability, of course, is completely different. So this little phylogenetic tree shows here SARS-CoV-2 variability pre-Omicron. So it's changed slightly since then, but I'm not sure if I can make this thing work. But this little tree over here shows you global diversity in SARS-CoV-2, and the panel at the bottom shows you the mutants in the global SARS-CoV-2 sequencing database pre-Omicron. So each one of those colored stripes is a mutation. And this is the situation with HIV. So it's kind of orders of magnitude different. From a viral perspective, it's an immensely more variable pathogen, and you can see that down there in that kind of multicolored bar, which shows you the mutants that characterize global HIV. So it's orders of magnitude different. I'm often also asked whether the mRNA platform is going to change our HIV vaccine design woes, and we have had plenty of woes, that's for sure. I don't think so. I don't think it's fundamentally going to get past the challenges that are associated with the HIV envelope that I tried to show you in the past couple of slides. But where I think it will really help, as you'll see, is in enabling us to test concepts a lot faster. The vaccine field's been hugely slow in testing new concepts, and I think the RNA platform that was so successful for COVID vaccines is likely to enable us to iterate really fast. There are definitely problems associated with the mRNA platform, and many of them are being addressed. So I'm not sure that it's kind of the silver bullet, but I think it's definitely going to advance the field. So what's the basis of current HIV vaccine approaches? The pendulum keeps on swinging in this field, as you may know, from T cells to antibodies to T cells to antibodies, enough to make you dizzy. We're back at antibodies at the moment, I think, but very much an appreciation that it'll be antibodies with T cells, which I think is still a problem in the HIV vaccine field, is that you tend to have people like me who only focus on one aspect, and very few people pull it together effectively. All the antibody-based vaccines are based on the fact that very rare HIV-infected people living with HIV, make what are called broadly neutralizing antibodies, which are capable of recognizing diverse global viral strains. So everybody who's infected with HIV makes good antibody responses, but only about maybe somewhere between 1 in 5 to maybe 1 in 10 make good antibody responses. And so those broadly neutralizing antibody participants are the people we're very much focused on for understanding how their immune systems work. Unfortunately for those people, and it takes about, this schematic down on the left-hand side shows you that it takes 3 or 4 years for those people who are infected with the virus to make broadly neutralizing antibodies. So even in the context of lots of viral replication and lots of time, even in that context, these antibodies are really rare. So to try to copy that process by vaccination is a challenge. And what's more is that even in those people who do make broadly neutralizing antibodies, they don't do any better clinically than people who fail to make neutralizing antibodies. In fact, often the opposite is true. That's a consequence of the fact that people who make broadly neutralizing antibodies often have very high viral loads, and that's why they made the good antibody responses, but it's too late for them. But the idea is if we could get those antibodies present before the virus infected those individuals, then that seems more likely to work. And there's now good evidence, which I'm not going to speak about today, from what's called the AMP trials, the antibody-mediated prevention trials, where people were given a broadly neutralizing antibody that had been made literally in kilogram quantities, and they were given it by infusion, 10 infusions over a couple of years. And while the trial itself wasn't deemed a success by the per-protocol measures, what that trial did show was that if people were exposed to a virus that was sensitive to the antibody that was in them, then they were protected. So it was the first proof of principle we had that people like me hadn't been wasting 20 years looking at broadly neutralizing antibodies, thank goodness. Because of that, and a long history of doing this work goes back 20 years, we've spent a huge amount of time as a field trying to understand the arms race between viruses and antibodies and studying those elite neutralizers in detail. And The technology's kind of gotten fancier over the years, but the basic question is the same, which is, how does the virus change over time? How does the changing virus force some people's immune systems to make antibodies, and can we copy that process to make a vaccine? And we now use, kind of higher tech approaches to sort out individual B cells and use those to isolate the monoclonal antibodies. We used to do it with plasma. It's the same kind of information that we're getting out of it. But it does, I think, provide us with a roadmap for HIV vaccine design from these unmutated common ancestors, these rare precursors of antibodies, and it provides us with a pathway for how they became broad. It is the basis of many current HIV vaccine strategies, and so I think the work has been useful, but there's a long way to go, as I'll maybe try and show you. So I mentioned that the vaccine field has gone really slowly. I think it's been a huge source of frustration to those of us in the field, to the world, how few concepts we've managed to test at scale in the HIV vaccine field. And so there's a push at the moment to do discovery medicine trials, which are much smaller trials, sort of in the order of 20, 30, 40 people, but to test concepts a lot faster, to test them using the bars that we have from the lab as a measure of success, and to test more concepts more quickly. And this is the basis of a new consortium, which is very much led by the SAMRC and the PI is Glenda Gray, who's also a proud Witsy, an IDORI member. It's called the Brilliant Consortium. It's the most contrived title for a consortium you will ever hear in your life. There were two of us competing for the grant. The other crowd were called Valiant, which seemed like a really good name. So we had to be brilliant in order to beat Valiant, and therefore we had to come with big ideas to realize immunization and laboratory innovation to end AIDS in Africa with new technology, anything to get into the word brilliant. And it kind of stuck. So this is the Brilliant Consortium. It's a USAID cooperative agreement. I've learned that a cooperative agreement means that USAID gets to say a lot in a lot of the meetings about the direction of the science. But the mission of the Brilliant Consortium, which is across Africa, as you can see from that map, is to really catalyze HIV vaccine discovery and an African-led vaccine initiative on the continent to develop an HIV vaccine that comes out of the African continent or doesn't. We're totally promiscuous on this. We'd take any vaccine that worked at this stage. But it's a multidisciplinary team from 8 countries across Africa. And I think I'm really excited about it. I think it has the potential to be transformative, and I'll try and tell you why. It was a year old last week. It's a 5-year grant, so that year seems to have gone really, really fast. But we had a kick-off meeting last year, and it's been a really active, engaged collaboration, which has been fun to be part of. It has kind of 4 overarching objectives, one of which, and the kind of, right up front, is the community program, and the idea that we need to partner with civil society to advance vaccine discovery. And so, for example, the monitoring of the grant takes place within the community leads, the community work stream, to try and get that engagement built into the program right from the very beginning. There's a clinical program which is aimed to design and implement early-stage clinical trials, and I'll tell you about our first one. The idea here is to build on vaccine candidates that we think will lead to broadly neutralizing antibodies. There's the discovery component, which is the third aim, which Abdullah and Patrick are very much involved in, which is to develop innovative preclinical HIV vaccine concepts. So that's everything from measuring the outcomes of those discovery clinical trials, which turns out to be a nightmare, to development of new vaccine candidates, to rolling out capacity for all of these things across the African continent to the partner sites. So it's a large and ambitious project. And then the last part is that element of collaboration and capacity augmentation. So that spans all the way from the community program through to the laboratory discovery program through to the implementation of the clinical trial programs. It's kind of a 2-stage approach. We were very pragmatic about this. We wanted to do clinical trials in the first year. And so we are using a first in Africa approach. We're taking vaccines that have been developed by others in the US, and we're putting them in novel combinations and testing them in novel populations. So it's a kind of a first in Africa approach here, which is very much aimed at kind of testing our pipelines, setting up all the analytic tools, making sure the clinical trial sites all function well, safety's good, all of those elements. And I'll tell you about the first vaccine trial, which hopefully kicks off in the next month or 2. And then the second part, which I'll speak to much more later, is integrating our own immunogens into that pipeline. The first Brilliant trial, which is called Brilliant 001, B001, is one of those first in Africa concepts. And here we are taking two vaccines which have been developed, one at the Fred Hutch and the other at Amsterdam Medical Center, both of which have gone into humans already and showed promising preclinical and clinical data. And we are stupidly, with hindsight, putting them together in all sorts of different prime boost combinations, and I'll explain a little bit the rationale. The reason I say stupidly is it turns out that either of these vaccine candidates is really hard to evaluate in the lab, and evaluating them together turns out to be technically really, really hard. But I think there's a good scientific basis for that. They're both what we call germline targeting immunogens, and that's based on a concept that comes out of those studies of people living with HIV, which shows that the precursors of broadly neutralizing antibodies are like vanishingly rare in our naive repertoire. So the precursor of an antibody that has the potential to become a broadly neutralizing antibody, I maybe have 1, 2, or 3 of them in my body. And so vaccine strategies really got to go in and selectively target those rare precursors. It's like looking for hen's teeth. There is a precedent for it, which was published last year, where they used a vaccine called EOD-GT8 and actually managed to expand those rare precursors to the point where we could detect them and we could show that they were maturing along the right pathway. But still only using, and this is where I was talking to you about sequences, still only using sequences to be able to measure success. These things are still so rare you can't pick them up in the sera. So we have a big gap there to drive them even further along the pathway to have neutralizing capacity that we can pick up in the sera. And that's what we're trying to do in this crazy trial, which is combining these two distinct germline targeting vaccines together. Either, you can see the top is the control arm. We just give one of those vaccines all the way along for four shots. The second group is a heterologous prime boost using one germline targeting and then a larger one to try and shepherd those responses. Arm 3 tries to boost even further by using a combination cocktail approach. And then the fourth one is the kitchen sink approach, and we're just putting every vaccine we have with every adjuvant we have into everybody at every time point. And that one, I think, probably has the largest chance of success because we have twice the chance of recruiting those rare precursors and hopefully more than twice the chance of pushing them along. So that's where we are in terms of the clinical trials. I'll speak a little bit right at the end about how we're going to measure success, but I want to spend the rest of the talk more about the discovery science, which Patrick and Abdullah are very involved in as well, which is integrating our own African immunogens into that preclinical pathway through to the clinical program. We spent a lot of time as a group talking about which parts of the HIV envelope we should be targeting. So that is the HIV envelope, that big blob is the HIV envelope. And each colored part of the HIV envelope there is a target of broadly neutralizing antibodies. So we once thought that these were really limited in terms of the capacity to recognize the envelope. Turns out that much of the envelope can actually be recognized by broadly neutralizing antibodies. But we've done work here at the NICD showing that the optimum combination would be three sites, the CD4 binding site, V2 and V3, which Mark and others will know well. V2 and V3 antibodies are much more commonly triggered in infections. We see that more often in natural infection. So we think they probably have less arduous maturation pathways. And so those are going to be very much the focus of what we're going to do. That and the fact that the CD4 binding site has been really well tackled by people like Scripps and Duke. So we figure that epitope is very well covered. And also based on the fact that work largely from the University of Zurich showed that people who developed broadly neutralizing antibodies had unusual envelopes, which were, at a sequence level, again, linked to their ability to make broadly neutralizing antibodies. And that suggests that the envelopes from people who made broadly neutralizing antibodies might be intrinsically good as a basis for a vaccine candidate. That's kind of the thinking for the brilliant immunogens. And so to be able to do that, we've been going back to all that work that was done looking at the arms race in the Caprisa cohorts over the last 20 years and saying which envelopes of those are associated with people who went on to develop good broadly neutralizing antibody responses. And we're using those now as vaccines to see whether they trigger the same kinds of responses in rabbits and then NHPs and then humans. We are focusing initially on an envelope that I have honestly spent 20 years talking about called CAP256, which was one of the first broad neutralizers we identified in the Caprisa cohort. And there are, we've done, I don't know how many papers we've published about this donor. She has been studied and studied and studied. And we know exactly which mutations were required in the antibody, and that's what's shown here, all the blobs here, to enable that antibody to become broadly neutralizing. And we know at really ridiculous levels of detail what happened to the virus to force the antibodies along that pathway. And so what we're trying to do now is to copy that pathway. We're taking the virus that infected CAP256 and using that virus as the prime, and then we'll incorporate into that vaccine a diversity that was associated with the development of broadly neutralizing antibodies in the donor, and do what's called sequential immunization in that way, which is a concept being widely explored by many in the field, definitely not just a brilliant thing. We focused also on CAP256 because we'd also shown that the envelope makes intrinsically good trimers. This has been a problem that's plagued the HIV field, is that many of these proteins just are wimpy and fall apart. This one's naturally pretty good at behaving itself, so that's another reason to use it as a vaccine. And it also is what we call germline targeting, in that we've shown that it intrinsically can trigger the precursors of lots and lots of V2 lineages in multiple different people. So it's like it's a imprinter envelope that is able to trigger a public response, and that's the kind of envelope you may want for a vaccine. We've been working with Roz and Patrick and Abdullah and Ryan and Wendy and actually many others to incorporate CAP256 into multiple different platforms. Much of the RNA work's been done here, and we're in the midst, we should get the next set of NEWT data today, actually, but we're in the midst of testing how effective that vaccine is at triggering antibody and T-cell responses. This is what the vaccination schedule looks like. So there's a CAP256-based envelope mRNA administered either with or without a gag to try and incorporate the T-cell aspects. And then there's an MVA booster arm. Group 4 was a long shot, which I think is not working out well, so I'm not going to talk about it anymore. And group 5 is the negative control. But we are now at week 20 and are testing those samples. These vaccines do seem to trigger good T-cell responses. I'm not showing the data, but that's from Wendy and Roanne. But they also trigger to, which we're excited about, strain-specific autologous neutralization after 2 vaccines. So these are the 5 groups again here, and these are essentially 2 negative control viruses. One is a tier 1 virus, which means it's extremely easy to neutralize, and generally you don't want that. And then MLV is a negative control, and you can see there's a little bit of background there, but nothing too scary. But in those rabbits who received the RNA prime and boost, we're actually getting really nice neutralization titers in more than half of the animals. So this is about as good as the neutralization data that's being generated by, for example, BG505 and other candidates in the field. So we think it's encouraging after 2 shots that we're getting those kinds of responses, and we will keep those going. I think what's even more encouraging is that they're hitting the right spot on the envelope. The whole premise of this vaccine strategy was that it recognized the top of the envelope. We wanted those V2 responses. And without boring you with lots of data, that's exactly what we're seeing here. If we introduce mutations into that region, we knock down the responses completely. So we're getting neutralization of the vaccine match strain, and we're getting neutralization that's targeted to exactly the right part of the envelope, and that's actually why we're most excited. Most of the neutralization seems to hit that spot, so that's good. So Abdullah's busy making more vaccines with variation within the V2 strand. So we'll see if we can broaden those responses to capture more breadth of responses. We're also going to do immunogenicity next in non-human primates, and we're going to compare the rhesus and the vervet macaques. Rhesus is what's normally used in the field for testing immunogenicity, but we haven't got any rhesus monkeys in South Africa, and they're generally, I think we have 12 in the whole country that are accessible. And so we're looking for another model. Vervet macaques, as anybody who lives in the country will know, are not rare. So the potential is to be able to use vervet macaques as an immunogenicity model, so we'll do head-to-head comparison then. We have already done some of that work. It does look fairly promising with another envelope, so hopefully that will help. And then we have an agreement with the Duke Vaccine Facility to manufacture clinical lots next year, which is completely terrifying to me because I think there's lots of steps we still need to do, and it's massively expensive. This lab rat had no idea how much these things cost. It's like $3 million to manufacture one clinical lot. So it's just crazy money. And then several other envelopes are feeding into the preclinical pipeline, and hopefully will form the basis of Brilliant 3. We're planning that this would be the third clinical trial. We have another clinical trial planned for the second year. I think it'll take that long to get there. So that's where we are. We're also doing, and this is the work I was telling you guys about, was we're also trying to do a lot more exploratory research to find more candidates that feed into the pipeline. The major challenges I've already alluded to with HIV is keeping up with the virus. There's a massive amount of diversity, and of course, almost all that diversity is in the envelope, which is the target of the broadly neutralizing antibodies. We have a better handle. A lot of the panels that we've used to evaluate HIV vaccines are viruses that are 20 or 30 years old now. And so there's a big push in the field at the moment to sequence more contemporaneous viruses and get a handle on what's actually happening now. The AMP trial that I spoke about, where we tested all those infusions of VRC01, had some breakthrough infections. And so we were able to look at those breakthrough infections, which are kind of from last year and the year before, and we have a better sense. But that's only in South Africa and in the Americas. The rest of Africa is pretty much a big black box still. And the reason that matters is that the virus is certainly changing. Antigenic drift is resulting in the viruses that are circulating in the population becoming more and more resistant to the types of antibodies that we are triggering. We had a sense that this was happening, and it's not a surprise. It's a consequence of the fact that everybody who's living with HIV is generating some of these antibodies, and so the virus is changing to accommodate those antibodies and then infecting the next person. But we didn't really have a good sense of how much it was happening, and there have been a couple of papers that have come out in the last couple of years, and one that we're writing up at the moment, that show that actually these viruses are becoming intrinsically more resistant to the types of antibodies that vaccines hope to elicit. So that again shifts the bar up again, because we have to trigger more of those antibodies, and they have to be more potent in order to neutralize strains that are kind of circulating right now. As I said, South Africa's a big black box. South Africa, but mostly the rest of Africa, is a big black box here. South and Eastern Africa, we have a reasonable amount of sequencing information, though we're not very good at mining it. And the subregions definitely have clear dominant subtype representation. This is a figure that Werner in Carolyn's group drew for me. And you can see the clade C dominance here in South Africa, the green thing at the bottom. But you can see, for example, in much of North Africa, it's O2AG, and we have very few sequences, as you can see from the top of those stacked bars. So we actually, and those sequences, I have to say, are generally pretty old. So we have actually no idea what's happening across the rest of Africa in terms of viruses that are circulating and whether the antibodies that we're triggering would actually work. There's a huge number as well of unique recombinant forms where the virus is recombining within people living with HIV, and we're getting even more of these kind of muddled viral subtypes, so we need to understand those. People like Gillian do lots of pol sequencing because people are looking at antiretroviral drug resistance. But for a vaccine perspective, those kinds of sequences are less useful, and we need envelope and gag sequences, and those are much less well represented in the sequencing databases. So a major focus of Brilliant is also to go and use the new network to find contemporaneous sequences and sequence those envelopes and gags, and then somehow, and this is why I was saying there's a problem with the data analysis, somehow take this huge amount of sequence data, and it is a huge amount of sequence data, and interrogate that to work out which regions we should be updating in all of our HIV vaccines. We've got nearly 500,000 envelope and gag sequences from 600 early or acute infections that came through several of the vaccine trials over the last few years. It's mainly subtype C, as I said, in the African trials, and B and BF from North and South Americas. But this needs to now be supplemented with all the sequences that we need to get from the Central and Northern African regions. And then somehow we have to mine this data in intelligent ways to be able to work out what we're going to incorporate in the vaccines. And we're not sure how to do that yet, which is why I'm looking at you guys. The other major part of the grant, as I said right at the beginning, is tech transfer and capacity strengthening. So I mean, African sites have been running clinical trials for forever. So running the clinical trial, I think, is less of a problem than the laboratory assays. Many of the laboratory assays are not in place in many of the labs that we are looking at. And so diagnostic labs, safety bloods, all of that, that's really well established in most of the sites that we're working at. But things like neutralization assays, BAMA, epitope mapping, the more refined assays that we will need for these kinds of trials are not in place in many of those labs. And so there's a big drive to transfer particularly the B-cell assays, only because they're much easier, which is why I do them. We'll transfer ELISA and we'll transfer the neutralization assays to all the different sites, partly so that the sites are set up so that they can do the vaccine trials, but also because the tools are really useful in the discovery research, so hopefully it becomes a sort of a self-sustaining pipeline. The T-cell preclinical assays, many people have been doing a lot of kind of standard ELISpots, but the more refined assays like ICS are less commonly implemented across the labs, and so Wendy Berger's at UCT is taking the lead on trying to implement those. And then we have to establish something called B-cell analytics, which is what I was referring to right at the beginning of my talk, and I'm nearly at the end of the talk, which is how do you gauge success in these germline targeting vaccines? I said to you, you can only gauge whether your vaccine has worked at the sequencing level. And that's a huge problem, because what that means is that you have to take leukapheresis from these people, and then you have to sort out the very rare B cells by B-cell enrichment. You have to pull out the individual B cells. Then you have to sequence the VDJ regions of the antibody genes in those individuals, and only then can you tell if your vaccine has really primed the right B cells. We can do lots of other work at the plasma level, but setting up the B-cell analytics would take us to the same point as the rest of the world in terms of germline targeting. When we wrote the grant, I thought my colleague Eunice Duarte in Kenya would do it, and then she said she was busy. So we have to set it up. It's proven to be enormously difficult. It's really expensive. It costs about $4,000 per sample. So it's a crazy amount of money, and we will have to do 40-odd individuals at 3 time points, really. So it's a huge amount of money, and just the setup requires hectic bioinformatics capability, which we probably don't have in place. A level of flow cytometry that we've never achieved before. So it's going to be very interesting. We have lots of support from labs across the world, so I'm hoping we'll manage. But we have to do it by June, I guess, because that's when the vaccine trial will be done. I hadn't really thought that one through. This is part of the capacity strengthening, is we've been doing site visits. The clinical team and the lab team have been going to sites, and I'm showing you some pics here from Uganda, from Kampala, and then we went to Kericho in Kenya, and Matera in Zambia, and those are 3 of the sites. The first clinical trials will happen in Kampala and in 2 sites in South Africa, Kampala, and in Zimbabwe. So it'll be a 4-study site. But you can see, there's a huge amount of goodwill in the network, and so hopefully it'll be a good collaboration. This is the network of partners and collaborators. You can see lots of people supporting us scientifically, but lots of funders coming in as well. The majority of the funding, as I said, comes from USAID, but we're leveraging everything we can at this point. This is a brilliant kind of leadership team, and it is diverse in terms of interests and diverse in terms of geography, and has proven to be a crowd of people that I didn't know before. They were not part of the networks I was part of, and it's been really the most wonderful thing starting to work with. Not without its challenges, I will say, not without its challenges to set up a new cross-Africa network, but really wonderful people. And then just to acknowledge my lab's funders, and again to say thanks to the team who have done lots and will have to do lots in the future. So that's me. Thanks.
I did want to ask about the vaccine trial that is going into human clinical trials.
Yeah.
That is targeting the unmutated common ancestors. Are all those immunogens targeting the same class of BNAP?
Yeah. So for the first clinical trial, yes, they both 46C and GT1.1, they're both VRCO1 class immunogens. And that's, I think, the class. So germline targeting is that pulling out the rare precursors thing, and you can have that for each of the different blobs on the envelope. The one that I think is likely to be successful is the VRCO1 class CD4 binding site, because it has such a constrained angle of approach that if I were to isolate a CD4 binding site antibody from everybody in the room, they'd all look the same. Like structurally, they overlap exactly, and that's why I think the VRCO1 class is probably the only class really that's amenable to germline targeting. The other reason is that if you're trying to prime and then polish, you want them all targeting the same part of the envelope, and then you want to diversify their glycan tolerance to make sure that they can get through the SMART-E sugars and be able to reach down to the peptide underneath. So both of those are VRCO1 class prime and boost. The one 46C is an engineered nanoparticle, so it's really twee, and then GT 1.1 is actually a trimeric construct, so that's why we're hoping it'll pull more lineages into the maturation pathway.
And then maybe just as another question, on your preliminary preclinical work that you've done with the CAP256,
Yeah.
so is that autologous neutralization
Yeah.
responses that you showed?
Yeah.
And despite being able to show that obviously there's mutations, you're targeting the area that you're interested in,
Yeah.
you're not seeing that breadth. Any idea why that's not occurring?
So I didn't actually show you the breadth data. We're not seeing breadth at this stage, though. So I showed you MLV and 6644, which is a tier 1 virus. So we don't want to see tier 1 neutralization. So that made me happy. We're not seeing neutralization yet of CON-C or CAP45. I think it's just too early to expect heterologous neutralization after just 2 shots. You know, if you look at Roger, if you look at the Scripps crowd, they're using 7 or 8 shots to get to that. I'm also not sure that just having autologous neutralization will get us to breadth, which is why we're incorporating the diversity, because I'm not sure. So at the moment, we've got the targeting and we've got the potency, but I'm not sure that that's going to overcome that barrier, and that's why we're incorporating mutations into the vaccines to try and force that diversification to happen. So we're trying to up the odds, but the odds are low, as you well know. Yeah.
Are we at a perfect storm now with the advent or advancement in AI to approach the kinds of massive analytic issues that you alluded to? There were several different streams of challenges that I wonder if AI could help with.
For sure, and is helping, not me yet. So people are using AI now already for several things. So for example, making those stabilized proteins. People are using AI to generate kind of optimally stabilized proteins. University of Washington's one of the world leaders in that, and they've got really interesting proteins that seem to be triggering really beautiful antibody responses. And those, are based entirely on AI. I don't think we understand. But the gaps are, in mining that sequence data that I was talking about, is how do we actually find the patterns in there. And in the B-cell analytics, at the moment, how we define whether an antibody response is good or not is based on 2 genetic signatures that are associated with those antibodies that I said all have the same classic angle of approach. But we're probably missing tons because we're looking, right at the one thing we know. So having a more agnostic approach to mining that data would be super cool. Also translating all of that into novel vaccines. So, there's tons of scope for clever data people, which I'm not one.
So, Penny, bearing in mind this comes from a TB researcher.
Always makes me nervous.
One of the most striking pieces of data you showed early on in your talk is the mutational landscape that HIV can occupy relative to another virus like SARS-CoV-2. I find that very striking, right? And so the question I want to ask you is, given the capacity of this virus to occupy such a large mutational landscape, is humanity constantly going to be seeking a new vaccine? Because you'll have a vaccine, it will last for a while, and then you'll have escape. Right, so that's the first question. What's your thoughts on developing ways that counter the mutation capacity of the virus? Right. The second is there nuance in the thinking in Brilliant and in the HIV field in general around where you're positioning the vaccine? So if you have a prevention of infection vaccine, you don't expand viral population and you don't have the same mutational landscape. But maybe a prevention of progression to disease, you That's a slightly taller ask. And so if you think about where you're positioning,
Yeah.
can you grab some low-hanging fruit? But it is really scary. It's like we're always going to be seeking a vaccine
It's
because
a
this
super
virus
amazing virus. Yeah.
just mutates.
Yeah. Okay, I'm going to have forgotten your second question after I've answered the first one, but I'll try. First question, it's definitely changing over time, but there are elements of HIV envelope that make it HIV envelope, and those are the sites that, for example, VRC01, this antibody that we're going after, still recognizes. It's recognizing it slightly less well over the time, but we're talking 20 years. You know, this is not happening like overnight. So we will need to update vaccines, but there are conserved elements of the envelope that, even in infection, come with a huge fitness cost. So if the virus can perhaps wiggle enough to get away from them, the virus is still going to be impaired. And so maybe at a population level, we might be able to drive those kind of changes if we had an amazing, wonderful vaccine. We don't yet have that problem. But we do have the problem of people who are not being diagnosed early and go on to have these chronic infections where they do mount these kinds of antibody responses that then put pressure on the virus. So if we could treat earlier and prevent that from happening, then we would also reduce the odds of the virus being able to get away. So it's, lots of elements to that. But there are parts of the envelope that are pretty conserved, and we have incredibly potent antibodies that can target them now. So if we could elicit something like that by active vaccination, we'd have a shot. And we are using, I haven't spoken about passive immunization at all, but we are using the products of that process, the actual monoclonal antibodies, very effectively now in kids and in adults to prevent and treat infection. Whether that will continue in the era of lenacapavir, not sure. The second question was?
Prevention of progression versus prevention of infection.
So HIV infection, that infection event is associated with a really, really stringent bottleneck. So if I was a person living with HIV who'd been infected for a very long time, I'd have millions of different viruses floating around me. But the infection event goes through a kind of a sieve, and literally 1 or 2 particles get across at that moment. So that's why that moment is appealing to me from a vaccine point of view, is because you have a much lower impediment. Those viruses, it's literally in most cases 1, 2, or 3 particles that cause the infection. The problem is that they are very diverse. So you still have to target the global diversity, but you maybe need less of the antibody. The other thing is that you want Fc effector functions associated with the antibodies to clear any local infection foci, and there's some data to support that. Therapeutic vaccination it's never going to involve antibodies. It'll involve T cells. But antibodies will never be useful as a therapeutic because the virus is just far too far ahead. So I don't see antibody-based vaccines as being useful for therapeutics. T cell vaccines, yes.
So going further, when you say, you won't be able to use antibodies on later on, those would be T cells. What's your view on drug-resistant patients? What can we do there?
So, I think the drugs, and you guys are more expert on this, yeah. But lenacapavir doesn't seem to have a easy escape pathway. They say none so far, which I don't believe. We've said none about many drugs over the years, no possible escape, and then the virus escapes. But I think the drugs are getting much better at limiting viral escape or causing such a significant fitness cost that the virus doesn't proceed. There are lots of cases at the moment where people are doing the ATIs, then so people are on ARVs, you do an ATI to release the virus and you put in a monoclonal antibody. And the hope is there that you stimulate enough expression of the HIV envelope for the antibodies to be able to zap the cells. And in many cases, that seems to be leading to increased rates of remission. So that seems to be more promising than I had thought, certainly because I have great faith in the ability of the virus to get away from antibodies. But they seem to be pretty good in non-human primate studies, and actually the work from Marina Kasky combining the most potent next-generation ARVs plus really good monoclonal antibodies plus latency reversing agents seems to look more encouraging than certainly than it did 2 years ago. We certainly see increased rates of remission in those people, but it's hard to do those trials.
So combinations of everything.
Combinations. It's the kitchen sink. I seriously believe it's the way we have to go, and I think it's the biggest problem in the HIV field. And in the vaccine field for sure, is that the folks at Amsterdam developed this amazing vaccine, and the folks at Scripps do, and the folks at Duke do, but nobody's putting these things together. And I do think we have a much better shot by putting all of them together into something where we maximize our odds. But the politics is just really special. And that's where I do feel we have an enormous amount of goodwill in this network. So I think we have a shot at moving the field forward there, because we've had commitments from all of the major manufacturers to give us the vaccines. So we'll see. We're probably the only people who can move it forward in the next year by combining them, fortunately, because everybody's being very supportive. But that, I think, is the biggest thing. And the same applies when you combine antiretrovirals and you combine monoclonal antibodies and you combine vaccines. We need to be putting them all together more effectively, I feel.
So can I ask something else before it goes there? Co-infected individuals, so with TB, I mean, are the participants that, because you mentioned placebos,
yeah.
So it would be nice to hear a little bit more about the placebos, and then how long will these individuals stay in the trial, and how do you look for the co-infection?
So we will avoid co-infection like the plague for the first in Africa trials. It'll be the most safe population we can possibly test it in. We're worried about HIV infection rates as well. It's phase 1 clinical trials will use people at very low risk. The placebos, it's 2 per arm. We'll have 8 per active arm of vaccine recipients, and we'll follow them for 42 weeks, 3 leukapheresis, and then we'll reevaluate. There's a possibility then to amend and add on another vaccine arm. But we'll see what SAPRA says.
Which didn't quite work as well as we'd hoped. I know that there are quite a few studies going on putting antibodies into infants to prevent vertical transmission.
Yeah.
And how much of what you've presented today can be used in that scenario?
Yeah.
So do you think that there's some differences in those?
Yeah, I left it out. Sorry, Jalila, I should have put it back in. We are using the monoclonal antibodies to put into babies at the moment. So we have a trial called PEDMAB, where we're giving babies who are born to moms living with HIV who didn't know that they were HIV positive, and we're giving 2 shots of the monoclonal antibodies as soon as we can after birth, and so far they look pretty safe. I think the pediatric population is still a gap for monoclonal antibodies, whereas I think adults, it's rapidly becoming a smaller window of opportunity. The peds, kids are smaller, so you need less antibody. It's also a really well-defined window of risk for the babies. So that, for me, is the one place where I really do see passive immunization playing a role. At a grown-up population level, not so much, honestly, because those antibodies are just too expensive. The other place that we don't talk about the babies very much is in the context of vaccination. Kids and teenagers who are living with HIV develop the most extraordinary responses to HIV, much better responses than we see in adults. And we don't fully understand why that is, but we think there are a few possible reasons. They have well-preserved CD4 counts and very high viral loads, so it's like the perfect storm for antibodies. They also probably have reduced tolerance stops. So many of the antibodies that get wiped out in adults may make it through in kids. So kids who are infected with HIV develop broadly neutralizing antibodies more times than not, compared to the 5% of adults. So vaccinating kids is and I didn't include it here, and it is very much part of Brilliant, but vaccinating kids is an appealing prospect. A long way away and would have to go through all the adult stuff, but I do think there's something interesting immunologically about that. So on the program, yeah.
Just have a question, because when I was watching the news, Prof. Salim Karim, when he mentioned that the new HIV vaccine that people take twice a year after every 6 months is 100%
yes,
protective against the virus. So my question is, on the general population that you guys used, like what were the most common side effects? Because I know a lot of people don't tolerate the vaccines.
So Slim was not talking about a vaccine there. He was talking about lenacapavir, which is a long-acting antiretroviral which you give by injection twice a year. Gillian, what are the side effects? It was relatively well tolerated. Steve, do you know? It was relatively well tolerated, I thought. Yeah, great results for sure. But I think the side effects were well tolerated by most people. But it's early, right? I guess we'll get a better sense of that. But that was a long-acting antiretroviral, so that's for a different use to vaccine and a different kind of biological principle. But that's what Gillian's going to say will make it really hard for us to test vaccines, because now we should be giving lenacapavir to everybody. And that means nobody should be getting infected. So how do we know if a vaccine worked? That's the complication. But it's a good complication to have.
Thanks for your always interesting talks. One of your earlier slides, I was just curious, it may be a simplistic question. One of your earlier slides showed that in Durban, the two sites, women were generally had a high incidence of HIV. Is there a biological mechanism for that, or a social mechanism? Does that have, if it's a biological one, does it have implications for vaccine design? Do you have to consider the differences between the sexes?
Yeah, so there are differences in the ability of men and women to respond to vaccines. I would say they're relatively subtle. So I would be delighted if we had a vaccine good enough for me to worry about that, shall we say. But there are some subtle differences. There's some really nice studies showing in many cases women develop very different responses to men. The biology of the actual transmission, we're very much dealing with the same thing in terms of a bottleneck with mucosal transmission. So there are definitely differences, but at the level we're trying to get a vaccine at, I think at this stage we're not even thinking about it, is the honest question.
So I'm sure modeling has been done to determine when this virus is going to reach error catastrophe, right? When you'll have enough mutational acquisition and the virus is no longer viable. So can you design vaccines that push the virus along that trajectory?
Yeah.
And then you have a time course within 3 years' time in the population. You're no longer going to be able to generate a replication-competent virus.
You're definitely a TB man. Yeah. No, it's a good question, and people are doing that. For example, we're testing adenovirus vectored vaccine that's going in now, and that's what they call the networked T-cell epitopes. And essentially, that's what it is you looked for they looked for the most conserved regions of the virus, and they try and focus the immune system on those to kind of force the virus into a corner. So far, this virus is not forcible into a corner. I mean, honestly, it's gotten past everything we've been able to throw at it. Maybe with the exception of lenacapavir, but I bet that will come too. That lovely idea of a ceiling, a mutational ceiling, just doesn't seem to pan out in the case of HIV. I think it's just incredibly malleable. But there are approaches to try and target what we call mosaic conserved regions, T-cell and B-cell approaches. We probably could do with some AI there. I'm hinting furiously here. Right.
It is interesting because one of the reasons multimorbidity is a real issue in South African population and clinical practice is the intersection between cardiovascular or other kinds of conditions with those who've acquired HIV, and then the ART, which for all the right reasons helps us all live longer. But that doesn't necessarily come into the vaccine design and program in the way you've described, quite understandably, actually. So just very generally and not related to that, could you reflect a little on the 7 or 8 settings, one in West Africa and then all the way down east to Southern Africa, and the relative complementarities or competitive differences? Because although strengthening capacities and single center efforts will, I'm sure, be key, I'm sure there's some that would span the different settings that you illustrated.
Yeah, it's something we've really struggled with, because when we thought about the grant, we really imagined that each site would have full capacity. And I'm part of another network called GEISER, which is a pathogen preparedness immunological network, and we tried that model there. It doesn't work. I mean, that we learnt in GEISER. We decided we would have B-cell sequencing and newts and ELISA at every site, and it was a catastrophe. So we're being much more grown up here, and we will have 2 laboratories, centers of excellence, who will do the first set of clinical trials. But we're trying to identify assays that are useful to others in looking at the viral diversity, because that's a big gap, but also in intrinsically measuring host immune responses, antibody responses, and using that to also address whatever the local pathogen of relevance is. So Lassa or Rift or whatever. So we're trying to implement platforms that are useful to the lab, and we've gone through a crazy audit kind of situation where we went to each lab, those were the site visits, and said what's actually going to be useful to you guys. Because trying to set all these assays up everywhere is not going to happen. I think there'll probably be, the B-cell analytics I think will probably always be based in one lab because it's just too flipping hard. But neutralization assays are easy, and people can use them then as the basis of a neutralization assay for SARS-CoV-2 or LASA or whatever, like we did. So that's kind of our thinking, is to be really strategic about what the gaps are. And also to ask the people in the labs what they want to be doing, because there is a funder tendency, I think. The Gates Foundation instructed us with Geyser that all assays were to be implemented at every site. And it was a catastrophe. So we've also, I think, got to be much better at pushing back on the funders and saying nope. And I don't think we're always very good at that. So we're trying to be a bit more vocal in what we think will work. But it's been, I must say, I have learned a hell of a lot. And less about the virus than I hoped, and more about other aspects of our life. So it's been really interesting, but we're trying a different model for the capacity strengthening. Yeah. Cool. Thank you very much, everybody.